thank Dr. Assmann and Professor Bar-Ilan and Shlomit Zuckerman, the Deputy Director of the initiative and one of my former doctoral students, for inviting me to come visit in Tel Aviv and contribute to this workshop. I also appreciate this beautiful specimen of genetic engineering that decorates the table. Old-style genetic engineering, I'm sure, but... Well, I have been thinking about these issues for, suddenly it seems like a long time. I started in 1990 when I went to the Genome Project to direct that LC program that you heard about a bit earlier, thinking about the downstream consequences of research on the human genome. As that project completed successfully, it was applied immediately, or at least aspired to be applied, to the practice of medicine, personalized medicine, as the rubric went. And I was interested in what that meant. There was a lot of rhetoric about empowerment and a lot of concern about privacy in that setting. So the sorts of issues that Oren sketched were our bread and butter in those years. In the last decade, we've switched now from personalized to precision medicine. It's an interesting rhetorical shift if you think about who runs precision equipment. Not amateurs, not patients, but professionals and experts. Now, from precision medicine, I'm seeing another shift in the field as it realizes that actually this is not about individuals so much. It's about groups. It's about stratifying people into different risk groups in the population and trying to use that to improve their health care. That, of course, when we talk about groups, makes us think about public health. And so in the US, we've launched a large precision medicine initiative to gather a cohort of 1 million Americans to contribute their DNA to a large longitudinal study. Talks about itself more and more in terms of public health rather than personalized medicine. So that's where this is going, and that's where I've now am going just because I continue to ride this train as it moves along. The last portrait down there is actually my great uncle, Ludwig Juengst, but I can see myself headed in that direction. So what happens when the Genome Project comes to public health and the public health initiatives? There's a little bit of a collision of worlds, and that's the crux of what I want to talk about today. It's looking a little bit downstream from most of the other talks in this workshop because we are not immediately talking about the privacy of human DNA. Now I'll get to that. But this is the collision that I'm thinking about. This, because both spheres, personalized medicine, genomic medicine, and the attempts to control infectious disease, in this case tuberculosis, have key features that are nicely contradictory, if you will. Leroy Hood, a personalized genomic pioneer in the US, likes to talk about it as 4P medicine. Genomic medicine is 4P medicine because it's predictive, personalized, participatory, involving the patients, and preventive, by which he means preventing the manifestation of diseases in patients who already have the germs, the genes, rather than primary prevention, which is the usual goal of public health, to prevent people from acquiring the germs in the first place. And as The rhetoric of personalized medicine is about individual empowerment. In the public health sphere, we're much more comfortable thinking about, overriding individual interests, including privacy interests, in order to secure public welfare, group-based, and for the most part, in TB control, retrodictive. We're trying to see where the outbreaks come from and what we can do to stem them. So that's what, this is the rub in my talk, is the collision between these aspirational values of genomic medicine as they've been articulated and the traditional public health ethos. My particular entree into this field came this spring when I was invited to go to Antwerp to work with Dr. Annelies van Rij, who's a TB epidemiologist. She and her team are excited to be on the trail of developing a new technique for TB strain identification. Tuberculosis bacteria come in many strains. There's a lot of families that have evolved around the world. And it's important to know which particular strain you're dealing with in public health interventions. So they would like to develop a technique for identifying and classifying strains of TB using whole genome sequencing of the bacteria. And they'd like to do it in a way that doesn't take 3 or 4 weeks to culture the bacteria in the lab before they can run the sequencing, but to do it in a quick, real-time way with next-generation sequencing machines that could do the sequencing pretty much, on the spot in a clinical setting. As part of their process, they thought they should think ahead to anticipate what the social impact of having this kind of tool might be. And so they put together a project to talk to a wide range of stakeholders, deliberative workshops to try to anticipate those issues. That's what I want to report on in this talk as a work in progress, far from finished. So I'll try to briefly sketch the background of how they think they might use this whole genome sequencing of bacteria in a public health way, and then tell you 3 stories from the field that exemplify the kind of issues that our deliberative process raised, the stakeholders raised, and then try to come back for some interim conclusions about what happens when you try to combine the genomic ethics with public health ethics. So, many of you will know this story quite well if you are involved in public health, but here it is. We start with a active case of TB. We've diagnosed it in a clinical setting, and we want to know what kind of TB. So genetics is very useful for that because each strain has its own genetic fingerprint, and you can decode it either slowly by traditional low-resolution methods using DNA markers, or in a very high-resolution way using whole genome sequencing. But that takes a little bit longer. With that information, you might be able to achieve the promise of precision medicine by coming up with useful tips for the treatment of TB. If you know that your strain has certain resistance to certain drugs, you'll know not to use those, for example. But for public health interventions, you want to go further, so you'll try to do traditional contact tracing to see who else in the community might have acquired that infection. This is where the whole genome information about the bacteria comes in handy, because you can locate your strain on the phylogenetic tree, an evolutionary tree of all these different families of TB. And that can give you some, not fingerprints, but footprints, genomic footprints of where these particular strains might have come from, where this patient might have gotten their infection. So the identification of the source, which would allow you to reconstruct the chain of transmission, which, together with the information you get from contact tracing, talking to people, might let you target future surveillance efforts more precisely. You know who the at-risk groups are within the community so that you don't have to waste time testing everybody. And perhaps with targeted real-time screening, you could even get back to the community to find undiagnosed cases of TB loose in the wild, as you were. So that's the process. What happens when you do it? First story is about the diagnosis of an outbreak, ultimately 29 cases of drug-resistant TB that occurred in multiple European countries, all traced back to the same strain and to a strain that was localized through this technique to a particular internment facility in Libya for African migrants who are trying to escape Somalia and Eritrea, and have hired these human traffickers to get them into Europe. Along the way, a bottleneck in the process was this camp in Bani Walid, which had atrocious sanitary conditions. That was the source of the infection. And the information, having this information, could have been quite useful. It was quite useful to the public health authorities, but it was also released to the media and the public. And almost immediately, politicians took advantage of it to say, see, we told you, it's all these migrants and refugees that are importing TB into our pristine land where we had successfully eradicated TB, endemic TB. Well, that's not true exactly, but that was the spin that they put on it. Just last week, the report was that the Belgian bus drivers have stopped serving Brussels North. So if you go to Brussels, you might want to find another station, because of their fear of TB and malaria and scabies. Well, really, actually, their fear of the transient migrants who are using that station to transit to the UK. Okay, what does this imply? On the one hand, good. It seemed that this technique seemed to work to improve, to provide a clue as to the origins of the outbreak and a suggestion towards targeted screening. Really, if you want to catch this strain of TB, we should look in those communities of asylum seekers that come through this route. On the other hand, look what happened when the information became public. TB is classically a stigmatized condition, and in this case, it compounded with the xenophobia of some people to stigmatize this particular group of asylum seekers as importing disease into Belgium. Well, that's interesting. Are these asylum seekers responsible for the European outbreak, or are they not? Or are they, in fact, victims of the outbreak themselves, having acquired this through really a socially determined cause, the funneling of their route through this internment camp? It also raises an interesting set of issues that phylogenetic sequencing underlines, and that is the ability to pinpoint the source of infections to very specific locations, like a particular facility in Libya. can trigger what the social scientists call the location stigma cycle. And this is briefly when a place like this internment camp acquires a reputation, notorious reputation for unsanitary conditions or other things because of some of the people who came from that place demonstrating these risks. That stigma generalizes to the location itself and then is passed on to anybody else from the location, whether or not they actually have the disease or display the stigmatized behavior or whatever it might be. Think of the various kinds of notorious places, bordellos, slums, neighborhoods, conflict zones, and the way that people get stigmatized just by having come from that place. Well, that's happened in the Bani Walid case and has implications not only for the asylum seekers' reception in Europe, but for their treatment, the treatment of their successors, the people coming behind them in the route itself, from local public health authorities or from their traffickers themselves. So it's a privacy issue, yes. Usually in public health, we're willing to suspend privacy rights and alert the public to risks because of the harm principle. We want to protect the welfare. But it has nothing to do with human genomics. It's a privacy issue that spins off of bacterial genomics. That's an interesting twist for people like me who've been worrying about privacy of human genes for so long. Here's the second story, a study, epidemiological study of the community around a prison in Lima, Peru, showed that as you move out from the prison in a circle of 5 kilometers, the gradient of risk of having TB goes down in a predictable way, suggesting that the source of the TB in this community is the prison. And in fact, they go on to show how permeable the prison is to the local community, with people going in and out all the time, visitors, staff, the convicts themselves being released into the community. The interesting thing that caught our eye about this was just the title. They called it Investigating the Spillover of Multi-Drug Resistant TB into the Community from the Prison. Well, one of the things that sequencing can't really do too well is to show you the directionality of a transmission chain. So they could have easily have framed this as a demonstration of the complexity of transmission chains in situations like this, where there are a lot of movement back and forth between a particular location and the broader community, i.e. as everyone's problem, not just the prison's problem. In this case, yes, the DNA mapping helped describe this risk gradient, but it was framed in a way by the public health authors in a way that imputed culpability to the prison, to the location. That certainly triggers this location stigma cycle again, and makes us think twice, I think, about the usual metaphors that we use in public health. The spillover from reservoirs of polluted water, the targeting of suspect groups, forensic, military, and environmental metaphors, which tend to underscore divisions within a population rather than connections. The fact was that this transmission pattern was determined, yes, socially by the interactions between the community and the prison. And so it could have been presented as a case in which, rather than dividing the community against the prison, we could have tried to engage the community in a solidarity-based approach to addressing the larger issues of the relationship. Finally, a third story. Summer of 2015, there was an outbreak of TB in Spain. Couldn't be resolved by traditional contact tracing. The patients just weren't connecting with each other. Until that September, 2 cases of TB were diagnosed with the same strain in 2 commercial sex workers. Oh, maybe that's why we couldn't connect the dots in contact tracing. Nobody was admitting that they got, that was their point of connection. That implicated the sex workers as the source and created a kind of public alarm and backlash against them. Because of that, the public health authorities did try to expedite the strain identification as fast as possible. And sure enough, using the whole genome sequencing method, it showed that their TB was unrelated to the outbreak within the community. It was, on the other hand, related to endemic strains in Russia, and so they were in that same situation of being immigrants importing the disease. And the clinical analysis also showed that one of the patients had HIV infection. Okay. Once again, you see some of the same themes. The improved strain resolution, in this case, the precision was helpful, was exculpatory in relieving the social stigma placed on these people, which was only compounded by the fact that their vulnerabilities were intersectional. It was a combination of the TB stigma and the stigma of commercial sex work. Of course, there were the public health response into the red light district in the city was typical of the location stigma cycle. But the new one, in this case, the new twist, was this incidental finding of HIV. Looking ahead to real-time phylogenetic sequencing, this could be a new kind of problem for the public health folk, because to do the real-time sequencing, essentially they take a sputum sample and do a metagenomic analysis of all the DNA in that sample. Well, that's any TB that might be in the sample, but it's also all the other microbes that would be in a sputum sample and human DNA. Uh-oh. Now we're back into the realm of collecting and analyzing human DNA with all the issues that we're familiar with in clinical genome sequencing of humans for clinical purposes. And the core issue there is, what should we do as public health officials with this extra information we collect when it might quite well be actionable, clinically important for the patients? Okay, you have TB, you also have 3 other concomitant infections. And oh, look in your own genes, you seem to have a risk for breast cancer. Well, ordinarily, the public health officials would just try to clean their sample as robustly as possible to avoid any contaminants, as they would have thought about it, and focus on the TB bacillus. Can they do that responsibly in this case, or do they now have a responsibility to look for other risk factors and somehow report those back to the patients when, in fact, this has all been done involuntarily and without the patient's knowledge in the first place? So if you look to the scriptures for advice, that is the secular bioethical scriptures, you can find lots of advice from both spheres. Here's a cover of a, I'm sorry you can't see it, a Presidential Commission on Bioethics report in the US on genetic privacy and progress in whole genome sequencing, which focuses on these questions of the return of incidental findings and the right not to know and privacy in those settings. On the other hand, you could also look to a framework for public health ethics and the emerging body of literature in public health ethics that tries to provide decision-making tools in that setting. On the first side, precision medical ethics, you do see the general ethos being very patient-centered, individualistic. The stress is on giving patients individual control over whether their genomes are sequenced or not, and how that information is disclosed, the privacy issues. When it is disclosed to the patient, there is an obligation to disclose results to patients, the transparency principle, and it means telling them all that you've discovered about their DNA. And of course, in the extreme, there's an obligation to protect patients from harms they might not know about by warning them about things they may not have even been interested in investigating. On the other hand, there's the population-centered realm of public health ethics, where we've traditionally said, in this case, private control and privacy are secondary to the harm principle. That is, people are free to do what they want until they begin hurting the interests of the community, and then we need to step in. That's bracketed, of course, by a second important public health ethics principle, which is, but in stepping in, we don't want to be draconian. We want to respect the interests and rights of people as much as we can, so you do it in the least intrusive way that you can, particularly when the groups you're talking about are themselves marginalized or voiceless. The rationale for mandatory screening and surreptitious testing in the public health setting has always been that, well, we all agreed to it somehow in the democratic process of setting up this public health system. So that's why we can do it. That's why we can invade people's privacy, because the democratic process authorized it. The only people that didn't participate in that process, of course, are the voiceless, like immigrant groups and marginalized communities that never had a chance to voice their view, even in theory. And so public health authorities try to treat those communities with a particular respect and lenience. Finally, in the public health sphere, the importance of social justice concerns of being fair in applying involuntary public health measures across populations. Well, how do these come together in ethics for precision public health? That's what we need to wrestle with. And I'll stop with just a few of our initial suggestions for that overlapping wedge of public health precision ethics. The stakeholders were public health officials used to tracking the flow of germs in that traditional metaphor through populations. This discussion suggested that really we should focus more on the social and structural determinants, the buildings, as Churchill was talking about, that shape the way this flow happens, that shape the course of these flows through society rather than the individual carriers. What is it that we could address in the larger arrangement of society that would prevent this? Secondly, the need to interrupt this cycle of location stigma by being as precise as we can about the actual origins of outbreaks, and by framing our descriptions as professionals in ways that don't exacerbate this kind of burden. Actually, the traditional way of naming variants in TB public health is by their origin, their place of origin. It's the Beijing strain. It's the Moscow strain, all of which feeds into this notion that there are notorious hotspots that need to be watched out for. Another unusual recommendation for public health was, if not, it sounds typical for other parts of biomedicine, was to actually engage the participants and communities in the decision making about what happens next. And finally, the one that probably got the most controversy from our public health laboratorians and friends was that, yes, maybe you should stick with your guns and ignore all that other DNA, since that's the least intrusive approach to doing this public health intervention, and the approach that focuses on the particular mission of these public health teams. Even though they said, what? Throw out all that information about other infectious diseases we might want to investigate? We're not just TB people after all. So I think there's interesting conversations to come about the intersection of genomic ethics and public health ethics as we go forward. And let's all listen and follow. Thank you very much.